I asked about the female because before and after 50, there is usually the menopause, and in another European project we found very different data in females across Europe before and after 50. The other point is if you have blood, because blood could be of interest for several possible biomarkers related to the gut microbiome and And did you check metabolites related to the microbiota? In the literature there are data, and we also produce some of them, that there is a relationship between the gut microbiota and products, metabolites, and what you can find in NMR in the urine.
Yes, I think the first question is part of the experimental design. So I think it's in the experimental design, but unfortunately I think we switched between, it was 55, the age group in the design. So we don't actually, I have not been looking into if there's an important effect of menopause.
We have also the age data, so we
yes.
can do it in
yes.
this kind of...
yes, but so far we have not done it.
No, so far we have not. The other thing is that according to your second question on blood, you know, how much was it the cost or the funding of your previous, so it depends on how much you have to devote to the nutritional part, to the technological part, to the economical part. So was a project of 3 million euros, so very, very small one. And we had a lot of partners because we needed a lot of partners. So you know that the recruitment of people for blood collection is completely different to do, and it must be done in the hospital. For urine is very, very different because you can ask the people to bring with them during the recruitment. So this is...
we would very much have liked to do blood, but also what we see in blood investigation is it's very often related to the lipoprotein, differences in lipoprotein profiles. But they are very good biomarkers.
Please.
Yeah, I have a more general question.
Could you come please to the microphone?
Okay.
Because we want to collect also the recording. Thank you very much. It's recording everything, right?
This was a really interesting session with all those presentations ranging from overviews to metabolomics and things like that. I'm here representing a more practical project, Higher Health Bread, and there was one conclusion, and that is a general conclusion that intake of dietary fiber is too low everywhere. Could be one interesting addition, I don't know whether you know that, are there specific, you can have intake of fibers from food, vegetables, whole grain, and maybe other sources. Are there indications in those fiber categories which is very low? And for instance, if people ate white bread and they change to whole bread, then you may change it in that way. So if there is specific information about the type of fibers. And the other thing is maybe a more difficult question. It was mentioned that butyrate is so important, especially for the elderly. And fiber is, of course, one of those prebiotics that can produce butyrate. Is there any insight in the project about which fibers and which conditions can produce more butyrate? Because that, if you know that, then you can maybe come to dietary recommendations for special fibers who do this in an excellent way. These are my 2 questions.
First question related to nutrition and dietary fibers intake. Of course, I did not go into details, but we analyze very much in details the food groups intake. And we do have, thanks to the very good harmonized data sets of Eurofir, we actually have very good information of dietary fiber content. Each food that was presented in food groups that contain the dietary fiber. So lots of additional analysis could be done. And I did not pull out some of the micronutrients that they are different between the affluent and ROP population. But also you have mentioned a very nice connection between the intake of fiber-rich food and also the gut microba. This could be some other fields of analysis that we can actually combine pretty well with metabolomics data.
Yeah.
And this is nice.
Yeah.
So we can do lots of analysis with actually the sources of fiber-rich food in each of the country.
One of the output of this project are information and is knowledge that should be shared. I agree with Søren that is willing as soon as possible to participate in international data banks for information. So we have a lot of metadata, a lot of information that we are willing to share with collaborations. I mean, so if we have people also feeling that could add value to our results, just sharing our data with them, these are much welcome. So this is the hope and the expected goal of this conference today is just to put together people that could be shared interest and share data to be analyzed together with different competencies. Enlarge the competencies that we have got with the 17 partners to even 100 partners if possible. So we are willing to do that.
I just add another question here, but just to add something about fibers, I think it's a very important topic, particularly in the light of data that Claudio presented about the reduction of microorganisms able to produce short-chain fatty acids in aging. So probably it could be a link. If you don't eat enough soluble fibers, probably you are not inducing the microorganisms that are able to metabolize them and to produce short-chain fatty acids. So it's a very interesting link. Sorry, please.
Yeah, I have a question for you. I wanted to know, you saw a difference between Finland and England, and I was wondering whether you used samples, urine samples, from old cohort studies. So whether there was a difference between storage time of these urine samples, which might explain the difference between the countries. Yeah, it's.
So you're talking about the storage time for the samples, urine samples. So the difference in that we have found. Let's say that we have a standard protocol for collection, urine collection.
So was it getting both collected at the same time?
They were collected and then stored at -80.
All right.
So it was frozen as soon as we got it in after 12 hours. So. Same time. So we stopped the time, I hope, at -80 degrees should be stopped all the changes in the samples.
No, but this was actually investigated. I think the main difference between UK and Finland is that it's completely different ethnicities.
Yeah.
Because in order to find people in risk of poverty in UK, it was Pakistani population which was sampled in UK.
The definition of ROP, say, at risk of poverty, is apparently the criteria are apparently more socio-economic. And so you have a definition of a risk based on socio-economic criteria. You assume that the diet is different, and then you compare. But why not to use, if you have more objective data related to body composition, to BMI, to something which is related not to an assumption of socioeconomic criteria. So I think that this is a very important point in your, but the number that you have collected is so high that you should have power enough to identify subgroup, even putting all together. I would like to put all together and restart the entire analysis with, for example, a machine learning approach to see what happened.
I think all partners agree. We assume that a population with low income had a different diet because literature suggested it. So, and now we also have anthropometric data, and so I agree. And probably an analysis, something like the, I don't know the name in English, the rete neurale artificiale, the artificial neuronal web. Yes, it would be important to analyze again all data.
Oh, this is great. So, I'm the first one to be heard by the audience. Okay, great. So I'm Claudio from the University of Florence, so I'm contributing the metabolomic study together with Søren. And I was very pleased by Søren's presentation. I would like to make a couple of remarks rather than questions. The remarks are the following. It's true that the initial assumption might have been optimistic or whatever, but I don't think this is a problem because we have all nutritional data, we have all, anthropometric data, and as Claudio was pointing out, it's a huge sample, it's a huge cohort. So it's a very valuable thing that we have put together all together. And furthermore, I'd like to say that the statistical analysis is actually not finished, and I think you agree that there are many more things to do. And of course, we started with the obvious ones, then we moved to something less obvious, and we are finding more and more interesting data. So I think there are reasons to be optimistic in the end that we will get good results from the analysis. The last remark I would like to make is that, as Søren told you, we were very careful in the way we collected the samples, in the protocol, and in the way we collected the NMR spectra and how we standardized the NMR spectra. So just let me say it because I have some experience in metabolomics and other fields, that this is a very good collection of sample and a very good collection, very valuable and very safe collection of data. So there is a lot that can still be done on this data. Last point is that, as you have seen, the data, the NMR data have been analyzed by bucketing the spectra, and this is the obvious thing to do, the thing that most labs do. But the spectra contain more information than that. And there are many groups, including Søren himself, our group, Francesco Capozzi in Bologna, in Cesena, and several other groups that are actually trying to extract, I said Bologna, but from the administrative point of view, anyway.
That you are from Siena.
Yes. From the administrative point of view, it's Bologna. So now the point I would like to make is that it is very likely that in the near future we can re-extract the information from the NMR spectra and squeeze even more out of it. And it's very good that the spectra are stored, not only the buckets for reasons of statistical analysis, but the spectra are stored, so they are stored both in Denmark and in Italy, and they will be there for a long time, so we can assure that. The samples is something more difficult to discuss, but the spectra will be there.
Alessandra from Biozone Food Innovation SME from Northern Germany. I'm very pleased that I participate here, especially in the first session that you were targeting some nutritional aspects regarding elderly, as we are coordinating in Biozone one project regarding personalized nutrition for elderly. And I noted here two questions, are more like general, for Mr. Claudio Franceschi. But before, I would like to congratulate for the great work performed in New Age. So it's more on scientific part. We are more on the SME, on the practical, and we see more, let's say, the income. First one is you were mentioning that a database gathering all the results generated from the study with the two groups of elderly that will follow the same nutritional for 1 year or 1.5 years. Will that database be open, or is an internal database in the consortium? Can SMEs maybe access these results?
The database has been is a modification of a NUGO database that was available to the consortium, so it's an improvement. And now we are putting the data in, and this data will be available to the partner of the consortium, of course. We have not yet decided if other SMEs outside the project they can have access to some results, of course, because Europe is asking us this, not to all the data, because we have to analyze and we are responsible for the data. So but in the final meeting, we will address this point. And if you are interested, let me know, because I can invite you to and be part of the final discussion and of the final decision of how to use for the next future the database and the data. So sorry, the answer is this one.
Yeah, thank you very much. This brings me to my next question. Were you targeting in the New Age project an immediate benefit for the SMEs in order to potentially increase their competitiveness, or that this will come more as a follow-up project after the New Age is finished?
No. They have to do a lot of work. We gave them a lot of money, so they have to do a lot of work within the 5 years of the project. Of course, there is a timetable, a Gantt chart, and they have to start just now, which is in the last 2 years, and we will have our annual meeting in Greece in October, and this is a major point to be addressed. No. The SMEs involved in the project has to produce a new prototype, some new food for the elderly. Yeah.
Okay, thank you very much.
No, thanks to you.
Is that an open meeting?
Sorry?
Is that an open meeting in Greece?
Is open we basically not, but we are thinking to have at least the final day open. Yeah, but we have not decided yet. I have to make a consultation among the General Assembly. But it's a good idea. Yeah, I would be in favor.
No, I was really impressed about biomarkers of inflammation. So we have some, it seems that we have some markers that could help us to follow healthness or to measure healthness or not, if inflammation is some kind of a property or quality of life, let's say, or related to the quality of life, especially if you say that aging is a disease. So I agree with you, aging is a disease, and we need to fight it. But it's just a provocation. But what I'm saying is very interesting to know that in blood you can find biomarkers, for example, fatty chains, acids, and we know that from beta-glucans, which are fibers that are proven to be are improving healthness. They are coming from microbiota degradation of these fibers, and so you can find anti-inflammatory molecules in the blood, and you mentioned other anti-inflammatories. Do you have, with this project, New Age, idea about what are the foods that, when eaten, that will be a source of inflammation or a source of anti-inflammation? That would be very nice. That would be a really first evidence that there is a link between what you are eating and what you are doing for your health. An immediate answer.
After answering, because my question is very similar, so you will answer all together. So other question? Okay, so I advise everybody to pay attention to Claudio's answer, to Francesco, and my question before having your lunch, because it could be very important. No, my question is simply, a nutritionist knows that the meal cause an inflammatory stress in the body. So we have to face a pro-inflammatory status at least 3 times a day. So what do you think is the impact of this continuous challenge during the lifespan on aging, I mean, and the question is the same. Which kind of meal could be less inflammatory?
So the answer is the following. The food contain a lot of most of the food is non-self. You eat, okay. So during evolution, the food are in a gray area between self and non-self, because are self, non-self which become self. And there is the oral tolerance, which is a mechanism very complex, in order to avoid that the proteins, for example, present in food do not become strong antigens. But still, the difference between an antigen and a food is in a gray area, is fluid, is liquid. So starting from this theoretical assumption, evolutionary speaking, the nutrients, when there is, as I said, for example, an excess of nutrient, the great discovery was that the excess of nutrients is sensed by sensors which are the same that evolution evolved to identify molecular pattern in bacteria. So there is no difference practically. Okay? So inflammation is a continuous. There is I skipped off a slide in which I said that there is an inflammatory tone, and that the first who used this hypothesis was Mechnikov. Physiological inflammation. So was the word of Mechnikov. So what is inflammation? Is nothing else than an increase with age because the body is bombarded by garbage. So garbaging is more broad than inflammation. Garbage is the number of a lot of stuff that I tried to make a list, but is very long, that are produced physiologically by our body, but during aging are produced more. And when you eat more food than necessary or a bad food, this is also another stimulus, okay? So within this vision, inflammation is physiological. It increases with age because there are more garbage. The physiological systems to get rid of garbage decline. So there is an increase in the inflammatory status. Inflammation is not a sign, a biomarker. Is a cause of aging, is a cause by itself. And is a cause because inflammation can cause senescence. Senescence can cause inflammation. Inflammation can cause senescence. And it propagates. We don't know how. We know that the blood for sure propagates. Aging. In the blood, there are hundreds of proteins that we are trying to measure. There are hundreds of compounds, but they are not simply biomarkers. They are something which maintain and propagate the aging process.
But they are mirror.
They are mirror, they are biomarker, but at the same time, not only biomarker, but active molecules. Okay? So, just to finish, the idea with New Age is let us know if a 1 year Mediterranean diet with the ingredients classical of Mediterranean diet plus fortification with vitamin D and so on is able at the molecular level, at the most advanced omic molecular level, what happened in order to understand if this hypothesis is true or not, to identify the mechanisms. Nobody knows the mechanism. We know that there are a lot of data that Mediterranean diet, but probably also Nordic diet, also Okinawan diet. Why not? They can be good way for health aging and health span, but nobody knows how. How? And we need this approach, this powerful approach, 2,700 samples, is an enormous resource that I would like to ask you to have the possibility to have access, because it's a tremendous work. I personally think that the distinction between the ROP and non-ROP and affluent is probably not there. But this doesn't matter, because you have data that you can resplit in different ways. So you have an enormous possibility. You can contribute a lot to the big problem. And of course, if you have some metabolites which can be inflammatory, please pay attention, because I think they would be of great interest.
Thank you very much, Claudio.
I will let you conclude, but I will say that just one thing.
Oh, sorry.
We were
Sorry.
we were asked, just an answer to him, because we were asked by the European community, is ROP diet less healthy than affluent diet? Then we answered, and then we have all data to do whatever the scientists like to play with. But we had to answer that. So the presentation from Søren was to answer the question made by the European community. The European community asked for that. And now we have answered that probably yes. No, it's more complex. But at the end, we found that affluent on ROP classification is also a source of variation. We cannot say now if it's better or not. We can say just that they are different in metabolomics. We are starting to say that. That's important, in my opinion. But I agree with you.
Okay. Well, okay. Okay, just one question, a comment for Professor about inflammation. I'm Neb Ilic from FINS Institute in Serbia. You are making connection like senescence and inflammation. It's like influence one by each other. Is that correct? But you don't have the proof for that, because yeah, but inflammation could be just a normal part of aging, because we are not forever. Normal part of aging. That means that we need to die from something, you know, and our body is responding. Of course it is causing diseases. That's not the point. The point is that inflammation could be part of aging process. And that will tell us every grandmother, you know, who has pains in the back or pains in the knees and so on, you know. So I cannot see like clear proof that inflammation is a direct cause for senescence, you know. I think it's probably all around. Okay, and the second is about diet and aging. So we had that famous experiment with C. elegans where they feed them on the age of starvation, and they live together. They live longer for 20%. So that could mean like in human life, we could live 120 years more. So that is also like one of the possibilities to study how inflammation is influenced by, let's say, with diet, which is on the age of the starvation. Because it's obvious that if we eat more, we have more problems, probably more inflammation and so on. And for the foods, a lot of fatties, they would be pro-inflammatory compounds because they are part of the inflammation pathway because of their chemical structure, you know. So that's just my comment.
Yeah, but I agree. It is demonstrated that postprandial inflammation is caused particularly by fats. So high-fat diets are much more pro-inflammatory than low-fat diets. And even high sugar, yes. And consider that the postprandial inflammation depends also on the calorie you introduce. So if you introduce less calorie, you have a lower inflammatory status. So probably one of the reasons why starvation is prolonging lifespan could be that people who are starving have less post-meal inflammation than, I don't know, but I think that more or less all data go in the same direction. So probably I think that the inflammatory role of food is something we have to consider carefully.